Welcome to the first webinar from Astrea Bioseparations. In this presentation, we look at the challenges in biomanufacturing and solutions we can provide to help you overcome these. Dr. Steve Burton, CEO of Astrea Bioseparations, will now take you through the presentation.
There is a downward pressure currently on the price of biotherapeutics, partly from reimbursement policies and increased competition, which is forcing manufacturers to look to more effective ways of increasing the productivity and production efficiencies. So if we look at some of these challenges, we've just listed them in bullet point form here. You can see that, as I'm sure you'll all be aware, manufacturing productivity and efficiency is the chief concern of the industry at the present. Some of the pressures that have been placed on the industry now, particularly through the likes of the emergence of the biogenerics, so there is now more competition in the industry. So we are looking to have improved methods for the purification of these products. But ultimately, the industry is looking to reduce its manufacturing costs and to increase the output of its manufacturing facilities in order to compete against alternative therapies and to meet the rapidly growing market demand for biotherapeutics. It's probably fair to say that downstream processing has really struggled to keep the pace with upstream developments and improvements in upstream efficiencies, and there remains a mismatch between the capacity of downstream and the capacity of upstream. And also, we're now seeing the development of new types of therapeutic products coming to market. In the recent past, monoclonal antibodies have represented certainly the major part, and still are the main part of the industry in terms of revenues and products on the market. But we're now seeing a growing number of new products coming on the market. And some of the platform purification processes that have been established, particularly protein A for IVIG, for antibodies, this isn't suitable, isn't appropriate anymore for some of these new therapeutic entities that are now being developed. So there's this requirement now for innovation and new purification solutions to overcome these manufacturing challenges. As I mentioned at the outset, many of these challenges that the industry faces can be addressed by use of affinity purification technology. And as many of you will be aware, affinity chromatography is not a new technique. It's been in use for many years. Originally, it was developed as a research tool and then became used in biomanufacturing. And more recently, it's become really the main centerpiece of many downstream processing methods. And this is exemplified with the use of protein A. So, as I'm sure you're aware, the majority of monoclonal antibodies today are purified and manufactured using immobilized protein A. And protein A adsorbents have really enabled the growth of the monoclonal antibodies into the level that they are today. So over 50% of the market for biotherapeutics is represented by monoclonal antibodies today. And to a large degree, this has been enabled by an affinity capture platform, a very effective affinity capture platform. However, whilst for monoclonal antibodies, there may be an existing solution, for other biotherapeutic molecules, no such solutions currently exist. And in this case, the benefits of affinity chromatography really can't be harnessed for some of these new products unless there is an effective affinity capture approach. So the ability to develop bespoke or custom affinity ligands that are specific for the target molecule of interest is really something that needs to be addressed. And this is important now for the development of effective purification processes for new biomolecules. If we just consider for a moment the types of affinity ligands that have been used in commercial manufacturing processes, that there's a number of considerations for the ligands that are going to be used in biomanufacturing. First and foremost, the ligands need to be selective for the target entity. They need to bind it specifically, and they, of course, should not bind the impurities that are present. They should be separated from the target. We also need to recover the target in high yield, and also we need to retain the activity of the target. And this is also very important. It's not enough just to bind the target effectively. We also need to elute it and recover it back again in high yield with high activity. The binding capacity is clearly important, as is the productivity of these manufacturing processes. And of course, the stability, because today these chromatography adsorbents are exposed to a wide variety of chemical and biological agents, particularly in relation to sort of cleaning of the adsorbents, for example, using sodium hydroxide. So the ability to withstand these treatments is important, as is the resistance of ligands to fouling. That they should not attract a large number of proteins or lipids or DNA that would impair their performance. Equally, for use in a GMP process for manufacture of a human biotherapeutic, that the ligand that's used, and indeed all components of the adsorbent, should not possess any toxic or mutagenic properties. Cost is a key factor, and increasingly so. The cost of the ligand can be quite significant. So having an affordable affinity adsorbent is clearly important. As is the production and the availability of ligands. And just to put this into context, it's not uncommon for an affinity adsorbent to require in the region of 10 milligrams per mil of ligand. That will be a reasonable level. If one translates that to, say, 100 liters of adsorbent, then we are looking at requiring a kilogram of ligand. And larger volumes of adsorbent and regular supply is going to require many kilograms of ligand in many cases. So the production and availability of ligand is important for the future as well. And there are many sources of ligands. Some of you will be aware of these in terms of existing binding compounds. Antibodies themselves can be used as ligands. A whole variety of biomolecule libraries, for example, phage display, antibody fragment libraries can be used. But the two areas I want to particularly focus on now for the rest of the presentation are peptide libraries and small synthetic ligands, particularly the ligands that we develop using techniques such as computational chemistry and high throughput screening. And back to our discover, develop, deliver message. It's true to say that today Astrea is the only bioseparations company that offers this combination of ligand discovery, adsorbent development, also with the ability to manufacture large volumes of chromatography adsorbents and supply pre-packed columns which have been produced to GMP standards. So that's a fairly unique offering that Astrea has. A little bit more information about Astrea. The company was established 30 years ago, over 30 years ago, and we have today an extensive range of separation products, particularly affinity adsorbents and pre-packed columns. Our main focus is small molecule affinity ligands, but we do have a significant manufacturing capacity now. So you'll see in some of the later slides, we now have the ability to make single batches of adsorbent up to 1,000 liters. And we have a manufacturing capacity today that exceeds 35,000 liters per annum. And a number of our products have been used in licensed processes. The majority of our products are manufactured to GMP standards. All of our production areas are clean rooms, they're controlled environments. And some of the applications of these products are quite large, and we have adsorbents packed into columns up to 2 meters in diameter. And more recently, we also offer semi-disposable pre-packed columns that really simplify and speed up the use of these affinity adsorbents. And in recent years, a total of 18 of our adsorbent products have been utilized in FDA approved or EMA approved manufacturing processes. So that's quite a significant achievement. The company has actually 3 locations, where our main base is in the UK with research and development in Cambridge, and our main manufacturing site on the Isle of Man, which is a GMP standard facility. So I now want to go through a few slides just demonstrating how we go about the discovery of new affinity ligands and the development of new affinity adsorbents. And that's really summarized on this slide here. So at the outset of a project, we need to understand what the goals are. The design specifications for the adsorbent need to be understood. And that then feeds into this quite well-established pathway now, where first of all, we employ techniques to discover a ligand, and having found a ligand that has the required selectivity, we will then move to further develop this into a usable affinity adsorbent, together with the process parameters, and then finally scale up that adsorbent to put this into manufacture and develop additional information that support the use of that material in a licensed GMP environment. So the first phase of this is ligand discovery, and the main focus here is identifying a compound that has the required selectivity. And this is just a first step in this multi-step process, so this is also important to keep in mind. Ligand discovery is not the end of the process, it's the beginning of the development of an adsorbent. So in terms of our adsorbent design and discovery techniques, we use a whole variety of methods. I'll briefly explain some of these here. We use extensively computational chemistry these days. So we're now able to create virtual ligand libraries based on a diverse range of compounds that we could choose to make. But we have chemical descriptors for these virtual ligands in our libraries, and we can then interrogate those libraries. We can use crystallographic information on the target, if that's a protein or a virus, to identify potential ligand binding sites for our virtual ligands, and we can use techniques such as blind docking or roaming techniques. And then, having found a potential binding site on the surface of a protein, and that could be an actual known binding site, such as a substrate binding site, for example, on an enzyme, or it could just be a region of the protein that will make a convenient site for the binding of a ligand to it. It doesn't have to have a natural biological function. Having identified a site, we can then use other techniques such as targeted docking, which enables us to perform virtual ligand screening. So today we can actually screen virtual ligands in silico and rank the calculated binding affinities. So that gives us quite a lot of information about the types of ligand that would have the potential to bind to the target and bind to the target at a predetermined location. So that's a very useful set of techniques, very similar to drug discovery that we have been using for a number of years now. And we can combine this approach with the more classical high throughput screening approach, where we make libraries of ligands and then screen them. And I'll explain in a moment how we do that. And we have two approaches to screening libraries. We can use more focused, targeted libraries that have been determined, essentially the output of the computational chemistry studies, or we can produce somewhat random general ligand libraries and screen these to see if any members of that library are able to bind selectively to the protein target that we have. And many of the more biological ligand libraries that are used tend to work on that basis, that there's no real predetermination of which ligand structures are going to bind. It's really relying on serendipity to find if a member of that library just happens to bind to the target with the required selectivity. So we'll explain in a moment how we do that. The ligand libraries, certainly in terms of the trizine libraries, we have well over 100,000 ligands in these libraries, and there's really no limit to the number of compounds that can be produced using the synthetic ligands. And in terms of peptide libraries, we work with small peptide sequences, typically trimers up to hexamers, and those libraries can be well in excess of 1 million peptides in those libraries. The screening process is generally quite quick. To identify a binding ligand usually takes in the region of 12 to 16 weeks, and at that point we will be able to provide target ligands attached to a chromatography support for verification of the purification performance. We have on this next slide now a number of ways, routes to discovery of new affinity ligands. If we already know of a ligand that it's capable of selective binding to the target, we can move straight away into the development of a custom adsorbent. And Astrea undertakes an increasing amount of work in that area, where the clients provide to us a ligand that they've identified themselves, or with the assistance of others, and then we will then immobilize these and to create a GMP standard chromatography support. If there isn't a known binding ligand, then we have a number of options at this point. If we have structural data on the target, if that's a protein, for example, then we can use that information to see if it's possible to identify binding sites. And if we can identify a binding site, then we can use these virtual screening techniques, these docking methodologies I've described, to identify potential ligands that would interact with that site. If we don't have any structural information on the target, it's certainly not the end of the world, because we can use other screening techniques to identify potential binding ligands. And again, I'll explain how we do that. So there are a number of options available to identify ligands, depending upon how much information is known about the target at the outset. So in terms of the small trizine ligands, this has been a very well established approach to developing ligands. These have been very successful, and there are many examples now in full commercial use, and this has been the case for a number of years. Some of the advantage of these small ligands is that they are small molecules, they are made synthetically, so it's relatively straightforward to make multi-kilogram quantities of this ligand. If you recall my comments from a few minutes ago about the availability of ligands, it's relatively straightforward and relatively quick to scale up ligand synthesis for these small types of ligands. That means we can produce very large amounts of adsorbent relatively quickly. And they're inherently stable, the small chemical entities, and they are certainly generally resistant to high concentrations of sodium hydroxide, and in many cases they're heat resistant, they can be autoclaved. And they have typically very long lifetimes as well in use, with really no measurable decrease in binding capacity, which is important over the lifetime of their use. And they also tend to be relatively inexpensive because of the simplicity of making the ligands. These are some of the reasons why these small synthetic or mimetic ligands have been of interest. We make them using combinatorial chemistry. I won't go into detail here, but it's very straightforward. We use a starting triazine compound that we can then decorate with various substituted amines in a very controlled way. And this enables us to produce a very diverse library very quickly. And unlike peptides and proteins, where it works typically with 20 or so amino acids, in the case of these synthetic libraries, there are many thousands of amine compounds that can be used. So that means that we can very quickly develop quite large libraries, and we're not restricted now to just 20 or so amino acids. We can use a whole variety of groups that can be used to introduce novel binding functions to interact with the target and the target binding site. Again, we synthesize these libraries using combinatorial chemistry. So it's very quick, very easy to do, and we can quite quickly make thousands of ligands for testing. Now, when I say we make ligands, it's important to understand that long gone are the days where we would spend a lot of time making a ligand using solution chemistry, attach it to a support, and then screen it. These days, we use solid phase synthesis to produce those supports, and that enables us to rapidly screen. And the synthesis that we perform, we're using established chromatography beads, in many cases agarose beads, so that what we're actually screening are not ligands as much as representative samples of affinity adsorbent, the ligands already attached to a chromatography support. And the screening we perform is not so much in a microtiter plate, but in a microcolumn plate that you can see here. We call these puriplates. And this enables not just screening for ligand binding, but we can also measure many other properties of the ligands, such as we can investigate binding conditions, how effectively different washing solutions might be, how effective different elution buffers might be. We can look into we can examine the recoveries from each step, the purities we obtained at each step, and we can even perform frontal binding studies with these microcolumn plates to give us a measure of binding capacity. So we can generate a surprisingly large amount of information at the very beginning of the screening process, using these immobilized ligands and this microcolumn approach. And in the diagram you can see here, that this sort of scatter diagram, this is actual screening data, and we're plotting here. These are from elution fractions from the plates, and we're plotting on one axis, on the vertical axis, just overall protein purity, and on the horizontal axis, we're plotting the concentration of the target protein. And what you can see here, you can see a whole bunch of ligands that are in the bottom left-hand corner. So these, the elution fractions from these compounds have a low amounts of total protein and low amount of target protein. Essentially, they haven't really bound anything. And in the opposite corner, the top right-hand corner, it's the opposite case. Those ligands are binding a high amount of total protein and a high amount of the target. So they're basically binding everything. The ones that are of interest are the ones that we've highlighted in the circle there. These have a much lower level of total protein, but a relatively high level of target protein. So that's evidence of selective binding to the entity that we're interested in. And we can further exploit this using multiple rounds of screening, where between each stage of screening, we will create a sub-library based on the information that we have on the structure of the binding ligands, and we can then screen these sub-libraries. And this will allow us to slowly but surely zero in on effective binding ligands. And I say slowly, this can be quite a rapid process overall. I mean, I think our record is something in the order of 9 weeks to identify a binding ligand, an effective binding ligand using this approach. So the other method that we use is to use peptide libraries. This is a slightly different approach. by using what's known as a split and mix technique, we can generate many thousands or millions of ligands relatively quickly. We don't know the structure of every ligand member in the library. These are attached to beads, but we can screen these, and then using a blotting technique by radio labeling the target, we can blot the resulting. We cast the resulting beads, the beads after exposure to the target, into a gel. We blot that gel onto a membrane, and we develop that gel and identify from the essentially autoradiography where the position of the binding ligand is, and we can compare that back to the original gel with the beads in, and then we can individually pick beads and subject them to micro Edman degradation to determine the peptide sequence. So this again is a fairly established method that we use. And using these techniques, we've been able to develop very effective affinity adsorbents. So this is just one example where we've used our trizine ligand libraries to discover a new affinity ligand for plasminogen, and that gives a very effective purification. It's over 340-fold purification of plasminogen from plasma, and that's in commercial use today for the manufacture of a therapeutic plasminogen product that's currently in late-stage clinical trials. So having found a ligand, we then need to move into the development of an adsorbent. And this isn't a trivial task because there are many steps involved here. And having found our ligand, we then need to identify the best way of attaching it to a support matrix. We need to optimize its attachment and its performance as an adsorbent. We need to investigate and understand the key variables in a sort of quality by design approach, so that we have a well understood adsorbent that has a very reproducible performance. So all of these activity needs to take place before we can move into manufacture. These are important steps to take. And moving into manufacture, this will be familiar to some of you from a GMP perspective, because what we're effectively doing here is replicating the scale-up of a therapeutic product. So we need to establish our equipment requirements, a user requirement specification for equipment. We need to identify the raw materials, the quality, the specifications of those materials. We need to be able to assay them. We need to have in-process assays to monitor the attachment of the ligand to the matrix, for example. And we need to have final assays to measure the performance, the capacity, for example, and other aspects of the functionality of the adsorbent. Having done all of this, we need to draft SOPs, batch records, validation plans, transfer those to our manufacturing site, and then formally validate the production of the ligand and the production of the adsorbent at production scale. And that could be anywhere from 1 litre up to many hundreds of litres of adsorbent product. And at the end of that, we will then produce validation reports, and we would have a GMP standard adsorbent that can be manufactured routinely in a very well-controlled manner. And that's clearly important for use in a GMP process. Similarly here, we need to generate additional supporting data if they're going to be used for manufacturing of biotherapeutics. So we need to generate information on the stability of the adsorbents, in particular, how well they maintain their performance and integrity when exposed to the various process buffers and cleaning reagents. Extractables and leachables are clearly very important these days, and we would always undertake these types of studies for our products. We would also develop shelf life studies, which are important. On occasion, we would undertake toxicities testing, although we've never yet, in over 30 years, we've never had a case of a ligand that we've found from either the trizine chemistries or the peptide approach, which has given any concern in a toxicity testing study. We would also need to look at identity testing, FTIR usually for this, for example. And in the case of packed columns, we would undertake shipping studies, the ISTA testing, to make sure that the columns retain their integrity during shipping. And all of this information will be compiled into regulatory support files for each product that would enable the user to then share that information with the regulatory authorities, again, to demonstrate these are well understood, well controlled materials. And just very briefly, as a indication of how successful these approaches have been over the years, we recently undertook a survey, and I think out of 39 projects that we'd undertaken, the custom ligand programs, we've reached our target objectives in 90% of cases, so a very high success rate. And in around 60% of cases, those adsorbents went on to be adopted by our clients. So again, a very impressive success rate. Of course, there's various reasons why an adsorbent may not be adopted by clients. Projects are sometimes stopped for various reasons, or the target molecule may fail in clinical trials and be abandoned. So it's not always the case that an adsorbent that's developed will go all the way through to use in a routine manufacturing situation, but that's certainly, we have a very good experience of that happening. And then finally, delivery. Having discovered a ligand, developed an adsorbent, it's really important that there's a sustainable manufacture and reliable supply of those adsorbents. The biologic manufacturing processes typically would be operated for many tens of years. And throughout that period, it's important these adsorbents are available with absolute control of quality and consistency. So Astrea has invested heavily in its manufacturing facilities, and we can now produce batches from 1 litre all the way up to 1,000 litre batches in our Isle of Man manufacturing facility. It's a GMP standard plant. These are not drug products, but we adopt the same standard of production, and we work to the ICH Q7, ICH Q10 standards that are typically applied to bulk active substances, drug substances. And we have quite significant manufacturing capacity now, not just for adsorbents, but also for pre-packed columns. So this is a photograph here of one of the rooms in our column packing suite. We have a suite of rooms which are ISO 7 clean room environments that we use for packing and testing our Evolve pre-packed chromatography columns. So today, Astrea has quite a wide range of products from simple ligand libraries that we now supply, adsorbents in various sizes and scales for use in manufacture, and then more recently a focus on pre-packed columns, either for R&D purposes, and in particular now our semi-disposable columns, the Evolve columns, and our GMP-ready pre-packed columns, the Evolve D columns and the Evolve S columns. And we have quite a wide variety now of what you might call off-the-shelf adsorbents, existing products that are used for routine use in manufacture of biopharmaceuticals. And we've now moved on to develop new platforms for new types of biologicals. And if you recall at the start of this presentation, I gave the example of how successful Protein A has been for monoclonal antibodies. Well, we've been able to develop other platforms now for other types of biomolecules, and this is just one example. It's the purification of albumin fusion proteins, and we replaced an established three-step process using a combination of ion exchange and an affinity step at the end with a more advanced two-step process with a very efficient capture step for albumin fusions, followed by an ion exchange polishing step. And the affinity capture step actually delivers a greater purity than the two ion exchange steps combined. And that's the reason why we're able to condense this down to 2 steps. And the performance of this is really quite impressive. So you can see here quite a number of different albumin fusion proteins that have all been purified using this 2-step approach. So to summarize, because of the challenges that the industry faces and this growing cost sensitivity, there is a need for improved purification approaches. Affinity provides an excellent way of doing this. Protein A has proved that, and we are now also proving it by the ability to develop new affinity adsorbents targeted at newer biotherapeutic products. And we're able to offer not just discovery of ligands, that's only part of the puzzle. We're able to develop adsorbents, we're able to undertake large-scale manufacturing of adsorbents to GMP standards, and also today we can now provide pre-packed GMP-ready columns to really speed up and simplify the use of these materials in biomanufacturing facilities. So you can see from this how we've now developed this discover, develop, deliver platform for that is enabling the development of new affinity adsorbents and able to solve some of the purification challenges that the industry currently faces. So on that note, I will end the presentation. I'll hand over back to Andy and invite any questions you may have.
Thanks very much, Steve. And we've had a few questions come in, so maybe I can start to run through those. One of them sort of goes back to your early slide, Steve, about some of the key challenges. One perhaps we didn't pick up on is about sort of environmental challenges and carbon neutrality or net zero. And this is becoming a key challenge in biomanufacturing. And Marcel was keen to know how Astrea are preparing for this and what are we doing to sort of support this sort of carbon neutrality. Is there anything you can comment on that?
Yes, I mean, that's a very good point, and it's an important point. So this is one of the reasons why many of our products are based on agarose. So this is a natural biopolymer. It's not derived from oil, for example. The process we use to manufacture our agarose beads, unlike the more traditional processes which use oil or a water immiscible solvent, we've developed a continuous process that's based upon a spray atomization technique, and that uses just water. So in the manufacture of our base agarose beads, we use nothing but agarose from seaweed and water. So I think that's one example of how we're reducing our carbon footprint, because we're not using oil-based products or solvent-based products to manufacture the base beads. And likewise, the synthesis, we can make kilogram quantities of adsorbent, of ligand, sorry, very often in an aqueous environment, in a quite intensive way. So we're not using very large volumes of solvent to make the ligands either. So these are some of the ways that approaches that we've been taking to reduce our carbon footprint.
Okay, thanks, Steve. Another question was, you mentioned about virtual screening. How well does that relate to the physical screening? Is there a good correlation between what we find in silico and in real life?
There is. It works surprisingly well, and we almost always find binding ligands from the libraries that we've designed from the virtual screening. So virtual screening is useful for predicting structures. We then make those structures and screen them, and we usually find that many of those compounds are indeed effective. And in many cases, they're not only effective binding ligands, but they also interact with the very same site that they were designed to target. So it's been very successful.
Okay. And you described the dye-substituted ligands. I mean, how do these differ from sort of common mixed mode ligands?
Yeah, so I mean, the mixed mode is these types of ligands have been around for some time now. It's really a combination of ion exchange and hydrophobic interaction, and they tend to be sort of relatively long-range interactions. You know, we're talking about surface binding here, rather than a targeted specific affinity interaction. So whilst one can use these simple trizine ligands one can decorate them with charge groups and hydrophobic groups to create a mixed mode ligand, that's not really the real power of these. The real power of these is to design these ligands to a specific site and it then becomes a selective affinity ligand, and that's very different to a mixed mode ligand. These are site-specific targeted ligands, and as I mentioned, by using this approach of decorating with substituted amines, there are many thousands of compounds that we can use there, that we can incorporate into these trisine ligands, and that gives us the ability to design ligands to pretty much any target or site.
Okay, fantastic. I think we've got probably time for one more. And fairly topical, this question. Various forms of RNA are gaining momentum, especially related to the COVID-19 vaccines. Is Astrea looking to develop affinity absorbance for these?
We are now actively developing new ligands and adsorbents, and hopefully you will see news on this coming out in the coming months. So we're targeting in particular viral vectors and virus type molecules, and of course many vaccines are of this type. So we're extending beyond protein targets now to vector targets and vaccine targets. And it's not just the adsorbents and the ligands that we're developing here. We're also, with our columns, for example, we're very proud of the fact that our Evolve columns, which have the ability to quickly repack them using disposable liners, that approach has been enabled some of the COVID-19 vaccine companies to very rapidly develop their products and quickly change out columns. And so we've seen quite a high demand for our Evolve columns from the COVID-19 vaccine manufacturers. We're very pleased with that.
Okay, that's fantastic. So thanks very much, Steve, and thanks everyone for listening. Just a reminder, as I mentioned, this is a bi-weekly event. And the next webinar is on the 18th of June. And here we're going to look at one of the products that Steve mentioned briefly. It's our multi-mode mimetic ligand library. And this gives clients the ability, or customers the ability, to do the initial screening themselves. And we've created a very diverse library where, there's 96 different ligands. And the idea is that you can do some initial screening and potentially get a hit where you've got a resin that to some extent binds either your target molecule or a target impurity. So the next presentation, 2 weeks' time, is from Dr. Patrick Gagel. If you do get a hit, what happens next? So hopefully you'll join us for that presentation. And what I'd also like to point out is, as I mentioned, thanks for attending. If you'd like to promote our webinars to your colleagues, then please use the hashtag Astrea Webinars. And for anyone that's doing that, will go into a draw and can actually win their own multi-mode mimetic ligand library. And we'll announce the winner on Monday the 3rd. So again, thank you for joining. We will post this webinar on our YouTube channel, and that can be accessed either directly through YouTube or through our website. So again, thank you everyone, and have a safe day.